we are recording. So this is our muscle lecture recording here. This goes with the same PowerPoint file that's available on Canvas. And again, this is muscle anatomy only. For those of you in the AAPHY 34A section, there's going to be a couple of muscle physiology lectures that go along with this. Muscles, when you factor in the physiological aspects, this whole system can get somewhat more complicated. So this is the lecture only. In the lab, we study individual named muscles of the human body, like the serratus anterior, the tensor fasciae latae, the extensor digitorum longus. We look at the origin, the insertion, the action of each muscle, and in some cases, the innervation. Okay, so this is just general theory. Now, let's get to it. So, the muscle's job is movement. We don't need Captain Obvious here to tell us that. We know that. However, muscles also function in what's called behavioral thermogenesis. Let's say you get really cold. When I was a little kid, we'd go in a swimming pool, and little kids don't retain heat that good, even if it's 75 degrees, 80 degrees outside, and all the adults are hot. A little 70-pound, 40-pound kid gets out of the pool, and we're shivering. That shivering is involuntary. You can't control it. You can't stop it. That's basically your brain saying, okay, muscles, we're going to die if we keep losing body heat. So shiver and muscle contraction generates heat. That's a thermogenesis, a way of generating body heat. So you get these really short, rapid contractions of voluntary skeletal muscles, only they're involuntary, you can't control them, and that's how we generate heat. Yeah, I've done my share of shivering at high mountain, on high mountain climbs, and a lot of dives off the Santa Barbara, Ventura Coast spearfishing. Okay, so muscle has 4 attributes that basically enable movement. Number 1, like me, it's irritable when I don't have my morning coffee. Well, I kicked the Starbucks habit a while back, so now I make my own, but I'm still irritable when I don't get it. When we say a tissue is irritable, it just means that it responds to a stimulus like a nervous stimulus from a neuron, maybe a chemical stimulus like a hormone or something of that nature. Now, number 2, when muscle does get irritated and it responds to a stimulus, it can only contract. It can only pull on something. It can only get shorter. So we say it's contractile. It actively generates a pulling force. It cannot push. It cannot twist, turn, or rotate. It can only pull on something. Number 3, muscle can actively pull on stuff, but at the same time, muscle, it's like this little hair scrunchie here. I stretch this out, and this is like muscle. I can stretch it out. It's extensible. Not everything can be stretched out. Try stretching out glass or porcelain or concrete. Those materials don't stretch. Ordinarily, number 4, it's elastic. Stretch it out, let it go. It snaps back to its normal shape. If you compress muscle, like say hyperflex a limb or something, and then reverse your movement, it'll go back to its normal, relaxed, preferred shape. So in this way, muscle is like a squeeze ball. You squeeze it and it goes back to its normal shape. It's like a rubber band. You stretch it out. It goes back to its normal shape. So we say it's elastic. Okay, so 4 attributes of muscle tissue: irritability, contractility, and elasticity, and so on and so forth. Now, all muscles are going to have these 3 elements to them. Every single muscle will have an origin. And on our models, diagrams, painted skeletons, origins are color-coded red. They're painted on the plastic replica in red. And then each muscle has the contractile portion that actually is where the physical force is generated. That contractile pulling force is generated by specialized muscle fiber cells in this belly or gaster. And then the muscle is going to have something that it inserts on, which is the more movable point. And on all of our plastic models and real specimens, those are color-coded in blue. Generally, an origin should be less movable and insertion should be more movable. So think about it this way: your vertebral column, your rib cage, not super movable compared to, say, your head or shoulders, which are more movable. Your shoulders are less movable than your upper arm. Your upper arm has a lower range of motion than your forearm and hand. So generally speaking, origins are closer to the mid-sagittal point of the axial skeleton and the thoracic, the vertebrae and the rib cage. Insertions are more distal and lateral to that, further out on the extremities, generally speaking. So here we have 1 and 2 muscles, a deltoid muscle, which is your shoulder muscle, and the triceps brachii muscle, just as an example. And these are attached to 1, scapula, 2, humerus, 3, ulna bones. So we note the sequence of bones from proximal to distal: scapula, then humerus, and then ulna. Scapula is less movable on the human body and skeleton compared to the humerus. The deltoid muscle runs from the scapula and clavicle to the humerus. So we will say that the deltoid muscle, your shoulder muscle here, you work that sucker out by, tossing kettlebells and, doing the right exercise with dumbbells. We can say that the deltoid originates. It starts on the scapula and it inserts, it finishes up, it acts upon the humerus, specifically the deltoid tuberosity of the humerus, which you now know about because we're done with the bones in this class. Okay, let's clear this. Triceps brachii is another muscle. This muscle has 3 origins, hence the term triceps. We have a long head, which goes to the infraglenoid tubercle of the scapula. The lateral head, which goes to the basically near the head of the humerus. And then we have an insertion down here on what's called the olecranon process of the ulna. Now, in 3-dimensional space, your upper arm does not have the range of movement of your forearm. Therefore, the parts of the triceps here that attach to the humerus, not just the scapula, are all considered origins. And just this part down here that attaches to the end of the ulna, the olecranon process, that is the insertion of the triceps. Okay, so we got 603 named muscles in the human body. And each and every single one of them is a complete organ, just like the heart, lung, liver, brain, spleen, pancreas, integument, and all that jazz. And each one's going to have an origin and insertion and action, or several, and a source of nervous input, stimulation, motor nerve, innervation. Okay, so we got to deal with the organization of a muscle as an organ first. Then we'll look at the cellular level of all this. So this is kind of a busy diagram here. Let's simplify this. All these textbook illustrations are cool, but it's such a thing as trying to say too much in too short an amount of time. So I made these diagrams to break it down. So if you think of a muscle, its construction is like a bundle of bundles of cables. Each cable is like an individual cell. So you take a bundle of cells and then you take a bundle of bundles of cells. And now you have a muscle. You got to wrap it out, wrap the whole thing on the outside. So a tendon is typically dense, regular connective tissue that attaches muscle to bone, bone to muscle. This tendon is going to expand outwards. It will be near continuous with the tendon, which itself is continuous with the periosteum of the bone. and it surrounds all of the contractile muscle fibers on the inside. This tendon will branch on the inside, and you'll have fibers of the same collagenous, dense connective tissue surrounding these bundles of muscle fibers. Well, the connective tissue that surrounds bundles of fibers is called the paramecium, and each one of these individual bundles is called a fasciculus, which is Latin for bundle. Fascicle in the ancient Latin language means bundle. Now, endomysium is loose connective tissue that surrounds an individual muscle fiber or an individual muscle cell. Okay, so muscle cells themselves, you can, well, I should say skeletal muscle cells. Not the same thing as all muscle cells or all muscle fibers. Skeletal muscle cells, they're monstrous. Those things are crazy big, as we saw in histology. Histology lecture 3 of 3. And each cell is multinucleate or senocytic, and that's kind of unusual. The only other cells in the human body I can think of that are multinucleate senocytic would be osteoclasts that break down old bone tissue with enzymes. So you can totally see these little purple nuclei lined up beautifully, and then you can see these nice striations here. Okay, so these stripes or striations that you see, this image in the background is probably easily 1,000, no, it's probably more like 1,400, 1,500 power magnification. If we were to blow this up to like 6,000 power magnification, you'd see the details of these little purple and pink alternating stripes here. And this is what you see. These stripes here denote the boundaries, the ends of sarcomeres. Sarcomeres are like an organelle, a functional unit of a muscle cell that generates the actual physical pulling force. And in physiology class, we spend, or the physiology section of this class possibly, we spend a lot of time discussing the sliding filament theory of muscle function and cross-bridge cycling and all that. But for the anatomy portion, no, we do not cover that. Okay, so these functional units of muscle cells, you see these little zigzaggy thin blue lines, you see that really thin sky blue line, and you see these thicker lines here. Those are all specific proteins. So if we look at this diagrammatically, we see we have protein filaments that slide past each other during a contraction. We have what are called thick filaments made of a protein called myosin, thin filaments made of a protein called actin, and the thin filaments sort of zigzaggingly attached to each other, as you can see, and this little point of attachment is called a Z-line. The Z lines denote the boundary of a single sarcomere. So we got one sarcomere here, I'm circling with the cursor, another sarcomere here, a third sarcomere right here, and I got half of a sarcomere here. There's this thin, faint M line that goes down the middle of this stack of thick filaments. Okay, if you look at this from a little bit less magnification, you may not see the individual lines here that well. You'll just see a darker stack of them. So where you have these thick filaments made of a protein called myosin stacking up, you have this darker line, and we call that an A band. A is an alpha or dark band. And then you have this thinner area composed of thinner protein filaments like actin, and this is called the light band or I band. Okay, so actin and myosin, the filaments slide past each other when a muscle contracts. A little bit of physiology to introduce here. It's actually the myosin that pulls on the actin, just like we might be pulling on a rope, say at a frat party at a tug of war over a big puddle of mud. Okay, so each sarcomere is shaped like a poker chip, What do you do with the poker chips when your winnings are big? You stack them. So you got these stacks of chips at your end of the table, and that intimidates the other players so that they're more likely to fold and you're more likely to succeed when you bluff. If your stack of chips is tiny, they're going to look at you and they say, I can bluff you up, boy. Or, just keep raising and raising until that stack of chips is gone, and then the player's gone, they're busted out. So from Z line to Z line, the Z line here is purple. Each outside face of one of these poker chips is purple. And then we see the actin thin filaments here in blue. The area they cover here is blue, and then the myosin thick filaments that make up the dark band or A band, they're in the center. So that's literal physical three-dimensional shape right there of an individual sarcomere. Sarco means flesh, mere means part. Again, I believe that's a Latin there. So you take bazillions of these poker chips, stack them end on end, dark band, light band, dark band, light band, just alternating like crazy, and you form a myofibril. So a single myofibril in a single muscle cell or muscle fiber. So fibril, myofibril, and muscle fiber are not NOT, not the same thing. Myofibril is almost like an organelle. So a myofibril is going to be inside of a muscle fiber, just like mitochondria, Golgi apparatus, endoplasmic reticulum, nuclei, and cytoskeleton will be inside that muscle fiber. So a really big muscle like your guns, biceps brachii, might have 100,000 of these things. Or I should say a single myofibril in a big muscle might have 100,000 of these things. Okay, so we have a skeletal muscle slide that I found in our room in SOS 103 before the lockdown. And I had my equipment. I brought my personal equipment to school that day, set it up, and was able to connect it to the big monitor on the wall there so you guys could see what I would see as I worked through your slides in my microscope and then gave them to you guys. You guys would work through the same slides. I'm able to get to about 2,000 power optical magnification. It's a high-end Carl Zeiss, made in West Germany, on good optics on that microscope. It's like 50 years old now, from 1971, but just think of it as like a 50-year-old Ferrari, ultra high-end for its time. So I can do stuff like this. So we went to stupid high power magnification, almost at the edge of the limits of what the laws of physics allow, without going to electron microscopy. And I was able to see dark bands here. And in these dark bands, you can see these thin, darker purple lines here. You can actually make out the M lines. And then we have the lighter green light bands of actin filaments and whatnot in there. If we'd had some different chemical stains, we might have been able to see the Z lines inside the green stuff. This slide on at low magnification was kind of bluish and gray. The green shows up due to chromatic aberration at high power magnification. We got some a sarcolemma here, the physical cell membrane of a muscle cell. And then we can make out some cell nuclei lined up here and there as well. So this is at crazy high power magnification, and we could actually see a lot of detail here. Now, some of the materials that make up muscle, when you think of muscle, you think of all protein. Muscles are just dense, heavy things made out of protein. We have a protein called titin, so-called because it's titanic in size. It's the largest protein known to the human race. It's about 34,000 amino acids, and its molecular weight is about 3,800,000. That's a huge molecule. And what titin does is it keeps your muscles from overstretching. If you overstretch a muscle, you'll cause the thick filaments and the thin filaments to disengage, and there's no way to get them back together again. When that happens, you cannot use that muscle cell. That muscle cell then wastes away and dies, and you permanently lose that capacity of your strength. Kind of like a video game where, you're taking punishment from the other side and your health is no longer green, it's now yellow going into orange. in a video game, we can go back and heal or respond. In real life, we cannot do that. So for that reason, it's very important to not hurt your muscles past the point from which they cannot recover. You want to hurt them a little bit if you're working out to build strength or get swole, but just a little bit. You don't want to overdo it because then, boom, guess what? You permanently lose that strength. It will not come back from a damaged muscle cell, muscle fiber. Anyway, titin connects the M line and the thick filaments to the Z line. Okay, nebulin is a big protein that's found in these thin filaments here, and basically it stabilizes these thinner blue lines here so they don't bend and flex and get all out of shape. Okay, so I have another PowerPoint which I'm going to pull up real quick. I believe it should be on the module for Canvas. We have these thick filaments made of myosin, and then we have the thin filaments made of actin. So we have something called the sliding filament theory. Let's go ahead and pull that up. What's going on out there? I hear the dogs yarking here. Let me just pull this up from my stick drive. Do a wildcard search. I don't like to, there we go. Sliding filament theory. I don't like to download stuff from Canvas. Let me stop share real quick and reshare. Here we go. So, sliding filament theory of muscle contraction. Whoops, there we go. We can sort of do that. We can demonstrate this like that. So, you just scroll down the slides. You can stop at any one point in time, and you can see how the two groups of proteins, thick and thin, slide past each other. And that is what happens on the molecular level inside a sarcomere as a muscle contracts. Whoops. So I'm pretty sure I have this posted to Canvas. Let's go ahead. We're done with this file. We don't need that. Let's go back to some other features of muscles. Gross anatomical features. And when we use the word gross, we don't mean it's icky. We just mean it's large enough. You don't need a microscope to see it. So we just got through microscopic muscle stuff. Now let's get back to normal size muscles as whole organs. So there's 7 categories of muscle by muscle architecture. Some use the word shape. I like the term architecture because shape just says, oh, it's triangle or shaped like a torpedo or shaped like a long strap. Architecture implies an internal construction, an internal organization. And there's more to a muscle than just the basic shape. It's how the fibers are oriented 3-dimensionally. That's kind of important. So I prefer the term muscle architecture. 7 categories: pennate, bipennate, multipennate, parallel, fan-shaped or convergent, fusiform, and sphincteral. So let's just deal with these in that order. Pennate muscles, or I should say unipennate muscles, they have their fibers arranged between 2 tendons. One of those tendons leads to the origin, the other tendon leads to the insertion. And the muscle fibers in between them that actually generate the contractile force, they're sort of lined up like the veins of a feather. These muscles have a lot of strength, high strength, but their endurance is not so good. And they have what's called a short excursion. And an excursion is the amount of distance a muscle can cover as it contracts. So you could have a muscle that's a foot long, but when it's fully contracted and only shortens to 11 inches instead of the 12 inches when it started, that's a short excursion. You could have a muscle with a long excursion. It starts out a foot long and it shortens down to 9 inches. It shortens by 25% of its length. That's a long excursion. Okay, so the peroneus brevis, also known as the fibularis brevis, which is one that I have illustrated right here. That's an example of a unipennate muscle. Peroneus longus up here is also a unipennate muscle, and extensor digitorum longus of the leg right here. All unipennate muscles. This is the lateral view of the lower leg, calcaneus bone of the foot, Achilles tendons, soleus and gastrocnemius muscles. Kneecap is out of the picture. So these are muscles off to the side of your leg. They're very important in fast, short duration, power delivering twitches that keep you balanced when you're walking, running, shuffling around, doing stuff like that. Now, bipinnate muscles, you have one origin in red, tendon leading to the origin in red, and two tendons leading, well, I should say two tendons leading to a possible insertion in blue here. I should have done the two, I should have flipped this upside down here, but I wanted to kind of keep the diagram the same as on this image right here. So these muscles, bipinnate, high strength, low endurance, a muscle with a short excursion. In the Eldar days of yore, when I was a kid, we also used another term, short pull. Short pull muscles are strong, but they have lousy endurance. Rectus femoris is one of your quads, one of the quadriceps femoris group, a fairly nice example of bipinnate muscle architecture here. Now, what determines the strength of a muscle is how many individual fibers it has in cross section. So on this photograph, it's like 1, 2, 3, 4, 5, 6, 7, 8, 9. There's nothing but a whole row of fibers along the entire length of the contractile portion of this muscle pulling on it. So that's going to give this muscle a huge amount of strength compared to other muscle types, like this sartorius here, which has much bigger, longer fibers, but there's fewer of them in cross section. Okay, now multipinnate muscles. Let me just double check something here. Okay, I'm going to shrink the video canvas of the Zoom toolbar down because it's interfering with this diagram here. Multipinnate muscles have a lot of tendons, and each tendon has a lot of fibers inserting on it from multiple sides, but they concentrate their force all on one spot, all on one insertion here. So we have a big old origin up here of the deltoid or shoulder muscle all along the acromion process of the scapula, and then onto about the lateral 1/3, distal 1/3 of the collarbone. And you can see that the shoulder muscle, the deltoid, is very effectively 3 different muscles that can do 3 different things in one single anatomical package. You work out your shoulder muscle to get it buffed up and strong, you're going to be tossing kettlebells in 3 different directions here to get all 3 portions of this muscle adequately developed, adequately worked out. Now, we don't really have any plastic models anywhere ever. Even the $80, $90, $100,000 Sindaver models don't show the actual 3D structure of this the way it is in real life, unfortunately. Okay, so, or at least they didn't show it before the lockdown. I was there with a Sindaver rep talking, and I mentioned it, and they said, nope, we don't show that. But the fact that he knew exactly what I was talking about indicates that other anatomy people had asked the same question. Okay, so deltoid muscle is our textbook example of a multipinnate muscle. Now, parallel muscle architecture, self-explanatory, all the muscle fibers, the contractile power-generating cells run in the same direction like parallel lines. Low strength because they have a low cross-section of fibers, but they also have really good endurance, and they have long excursions, so we can call these long pull muscles. 2 premier examples are in the thigh. Gracilis muscle here, as I'm highlighting it with the cursor, and sartorius muscle here, the longest muscle in the human body that I'm highlighting here with the cursor. If you're trying hard to get washboard abs, doing sit-ups and crunches, your rectus abdominis muscle is another example of a muscle with parallel muscle architecture. Now, convergent muscle architecture. In the elder days, you could have called this deltoid muscle architecture, but we want to nerf the term deltoid, make it go away forever, and not ever bring it back for a reason. You can also call this fan-shaped architecture. So you look at this, a broad origin, and the fibers all converge into a narrow insertion, so the whole thing looks like a fan. Pectoralis major is a great example. The origin of the pectoralis major all up and down the sternum and even into some of the costal cartilage ribs here, and even part of the collarbone, the proximal or medial half of the clavicle. But it all gets concentrated on one spot over here on the humerus. So it's fan-shaped or convergent. Why do I not like the term deltoid? Because delta is the shape of a triangle also. There's the deltoid muscle right here. Now, the deltoid muscle does not have deltoid convergent fan-shaped architecture. The deltoid has, as we just mentioned, multi-pennate architecture. That's why I'm very happy to see this term go away, fade from existence, and go into the same dustbin of history that the dinosaurs are in, because it's confusing. And we need to make anatomy and science and engineering and stuff not confusing, less confusing, not more confusing. Okay, fusiform muscles are sort of shaped like a torpedo or some of those really fast ocean-going fish, or the fuselage of an aircraft. You know, expands in the middle, but it's narrow at either end. Individual muscle fibers are thicker in the middle than they are on the end. So a good example of muscles with fusiform architecture: biceps brachii and triceps brachii. I have an old textbook that stated that the biceps was a bipennate muscle. That is not the case. I guess all contemporary information says the biceps and triceps in your upper arm, your guns, are fusiform muscles. Not the strongest, not the weakest, not the best endurance, not the worst endurance. Okay, now there we go. Last are what we call sphincteral muscles. Sphincteral muscle architecture, we have fibers that enclose an opening, and in many cases you could argue there is no origin, there is no insertion in the strictest sense. There are just two areas where the muscle attaches to either soft tissue or maybe bone on one side, soft tissue on the other. So generally speaking, when you're dealing with sphincteral muscles, you don't have a bone as an origin or an insertion. It's just the skin and other soft tissue things like tendons or ligaments. So we have what's called here a band of connective tissue called a modiolus. It's on either side of the mouth. Your typical textbook doesn't use the term modiolus, but other internet sources that are more specialized in this area do. Orbicularis oculi surrounding the eyes, orbicularis oris surrounding the opening of the oral cavity, the mouth, are sphincteral muscles. Now, that being said, what I'm highlighting here with the cursor, the muscle of the upper eyelid, that is called the levator palpebrae superioris. That really is not sphincteral. It's often considered separate nowadays from the orbicularis oculi here. You use the levator palpebrae muscle to blink, open or close your eyelids. But if you're squinting, because say you're driving westbound on the 91 freeway or the 105 freeway, you know, let's say you work in Norwalk or the OC, Orange County, and you live in Inglewood or Hawthorne, you're driving home on a 105, 91, and the sun's setting right in your face and your windshield's dirty, you squint because it's hard to see. That's when you're using the orbicularis oculi. Okay, so we gotta go now because I haven't eaten food yet today and it's 2:17 p.m. We already covered biomechanics. I decided to cover biomechanics right after arthrology. Technically, the nervous system is supposed to be next. Okay, so that's it for this. Stop share and stop recording, and I'll get this posted up to Canvas shortly.